the 2022 Nicholson Prize, which is awarded to early career investigators in human immunology and vaccine research. And today, Brittany is going to tell you about her work on training anti-cancer immunity against pancreatic cancer using albumin-hitchhiking vaccines to recall ancient immune memories. Brittany, thank you.
All right, thanks, Yoji, for the introduction and also for the invitation to present my work today. I'm excited to be here. So I started my lab here around 4 years ago. In 2021, I joined University of Minnesota. And my lab works on immunoengineering. So we work on training the immune response. I actually started my work, as Yoji mentioned, working on tolerance for autoimmunity. And then during my postdoc, worked on mucosal vaccines for infectious diseases. And then just in the last few years I've started pivoting and trying to apply some of those engineering targeting strategies to cancer. And I think there's really a ripe opportunity to use some of these targeting strategies, especially for mucosal cancers, such as pancreatic cancer and others in the gastric tissues. So today I'll be telling you about how we are trying to train anti-cancer immunity against pancreatic cancer using albumin-hitchhiking vaccines to recall ancient immune memories. And pancreatic cancer, as well as other gastrointestinal cancers, present a persistent threat. Pancreatic ductal adenocarcinoma, PDA in particular, has the lowest 1 in 5 year survival rates of any cancer. In fact, only 12.8% survival 5 years after diagnosis. And this poor patient prognosis is due to a few reasons. One, surgical resection usually isn't a viable option and is not effective. Chemotherapy responsiveness is not great. Again, median survival only less than 12 months. And despite progress that's been made against other cancers with immunotherapies like checkpoint blockade and CAR T cells, other adoptive cell therapies, that really hasn't been effective in PDA. Pancreatic ductal adenocarcinoma has just not been responsive. And one of the reasons for that, a main reason, is because there are just too few immune cells in the tumor environment. PDA is characterized by a highly dense, fibrotic, immunosuppressive tumor microenvironment. So what this means is it cripples the adaptive immune response. Immune cells can't get in. Those that are able to get in are highly immunosuppressed. But then uniquely, the pancreas, given where it's located anatomically, nestled among all these mucosal tissues around the gut, also near the liver, it's a unique challenging for drug delivery as well. It's very hard to get drugs there and to overcompensate for this tolerogenic bias of these tissues. And so our challenge and our goal is to use engineered vaccines or immunotherapies to wake up the immune response in pancreatic ductal adenocarcinoma and other immunologically cold tumors. How can we wake up the immune response and reverse this immune desert? And where better to wake up an immune response than by targeting the place where immune memories are made? Does anybody know what this is? I don't know if this is usually an interactive seminar, but so this is a cleared microscopy image of a mouse lymph node. And the firework bursts on here are germinal centers within this lymph node, and they're lighting up because this lymph node is taken from a mouse that was immunized with a lymph node targeting vaccine. And we now know from the last about 10 to 15 years of infectious disease vaccine research that if we can design vaccines to, one, target to lymphoid tissues like lymph nodes where adaptive immune responses are orchestrated, but then take it one step further and target vaccines to specifically germinal centers within those lymph nodes, we can activate a stronger immune response, particularly a stronger antibody response, and also reduce off-target effects. So we can make a more effective and safer vaccine. And these germinal centers, these firework bursts here, they present an attractive target because they serve as a training ground for a high affinity antibody response. And as mentioned, these are the signaling hubs where immune memories are made. So out of a germinal center response where germinal center B cells and T follicular helper cells go through rounds of antigen-specific signaling back and forth. Out kicks from that plasma cells and memory B cells, which can generate these high affinity antibodies. And while, okay, antibodies aren't usually what we think of when we're talking about cancer. We think about them a lot in infectious diseases and infectious disease vaccines, but they've been, I would say, relatively untapped for cancer. So why would we try to target this site where immune memories are made for an antibody response? Well, we're trying to target the site where immune memories are made because we're trying to wake up the immune response by recalling ancient immune memories. And these ancient immune memories are endogenous retroviruses that actually were part of viruses that infected our ancestors millennia ago. So many, many millennia ago, our ancestors were infected with these retroviruses. They incorporated into our genomes. And then over all of these years, they persisted and now are present in our dark genome, which is actually the majority of our genes. And typically they're silent in normal tissues. They're not expressed, but we find that they're errantly upregulated in tumors, including tumors like those found in pancreatic cancer. And so this makes an attractive tumor-specific antigen target. We find them upregulated in pancreatic tissues around tumors. And in particular, what's really exciting about this as a vaccine target is that we find that in both humans, patients, and in mice that develop tertiary lymphoid structures around their pancreas tumors with germinal center responses against these endogenous retroviruses, they experience better outcomes and survival in cancer, in pancreatic cancer. So this work came out a couple of years ago demonstrating that these antibodies against endogenous retroviruses could lead to better survival. This was in lung adenocarcinomas in both mice and humans, but specifically they found that when these tertiary lymphoid structures, which are like a transient version of a lymph node that form around a tumor to activate an immune response, we kind of can think of them as a field hospital. When these form around a tumor, and when we see the presence of activated germinal center responses here, specifically against endogenous retroviruses, outcomes are improved. So we thought, can we use this as a target for a vaccine, targeting these endogenous retroviruses and targeting germinal center activation to aid in an anti-tumor response? And as we started looking more into this, we found more examples actually of this coming to light, where germinal center activation, engaging a humoral B cell response, and particularly targeting endogenous retroviruses can be effective against cancer. And the reason improving survival outcomes. Again, this is just kind of a schematic here showing a tertiary lymphoid structure, this field hospital where we have aggregates of immune cells, the training ground for high affinity antibody response. And why does that lead to better outcomes? It's thought that it engages natural killer cells to aid in the anti-tumor attack via antibody-dependent cellular cytotoxicity, or ADCC. So activating these B cells and then tagging tumors subsequently with these antigen-specific antibodies against ERV engages the NK cell response, activates those NK cells. And we see specifically in PDA that higher NK cell infiltration into tumors corresponds with better survival. So this was our goal: wake up the immune response, activate anti-tumor immunity, pulling in these other branches of the immune system via humoral immunity. And my PhD student, Noah Sinclair, is the person who has been taking on this project. We work closely in collaboration with Paolo Provenzano and also Lou Mainsky on the ERV aspects. And our first goal was, can we deliver a vaccine specifically to peripancreatic lymph nodes and lymphatic tissues draining the pancreas? Because the first step is, well, we want to activate immunity proximal to the pancreas in this region. And oral administration or an oral vaccine is an attractive route here because lymph node sharing is caring for pancreatic immunity. We see that there's a shared lymphatic axis between the pancreas, the liver, and the gut. This was I thought, a really cool cover that came out, I think, 2 years ago on immunity, showing this shared lymphatic axis and the drainage pathways between the liver, the pancreas, and the gut, illustrated like the L in Chicago. And so our goal here, our second question is, not only can we target an ERV-specific germinal center response to activate anti-tumor immunity, but can we do that by developing a vaccine that exhibits enhanced uptake in gut tissues in order to target these peripancreatic lymphoid tissues via this shared lymphatic axis? Okay, so from here, I'm going to shift gears a little bit, lay the foundation for why we're working on this type of vaccine strategy for pancreatic cancer. And I'm going to take a step back and talk about the vaccine engineering and the platform that we originally developed. I mentioned that we first were working on this for infectious disease vaccines. So, well, why would we want specifically mucosal vaccines against infectious diseases? We know that transmission of the majority of infectious diseases takes place via mucosal routes. And thus, rational vaccine design would be to activate immunity in mucosal tissues of transmission as a front line of defense. In contrast, those vaccines that we typically receive in the clinic, those that are injected, which we call parenteral vaccines, injected subcutaneously or intramuscularly, actually don't typically activate immunity in mucosal tissues. Rather, they activate immune responses in the blood. And this can be very effective for protecting against severe disease and death, for example, in the setting of COVID, but it usually doesn't protect against infection and transmission. And for other more challenging viruses such as HIV or tuberculosis and other pathogens such as tuberculosis, we really need these other immunization strategies that can activate mucosal immunity. And we know that in contrast to injections, applying vaccine components topically directly to mucosal tissues can be an effective way of activating this type of mucosal immune response. And it activates mucosal immunity by targeting mucosal-associated lymphoid tissues that underlie the mucosal barrier. So we talked about lymph nodes in the introduction and how they're the site of antigen-specific immune responses and where adaptive immunity is orchestrated. Well, mucosal-associated lymphoid tissues are the analog to that, just directly underlying a mucosal barrier. And the really cool thing about the mucosal immune system is that we think there's this network of common mucosal immunity, where if you activate an antigen-specific immune response in an inductive site like the nose, it actually triggers homing of activated immune cells from that site to other distal sites, including not only priming it then in the nose, the upper respiratory tract, but also trafficking to the lungs, the lower respiratory tract, the salivary gland, and even the genitourinary mucosa. It does this through expression of homing markers on activated B cells, T cells, plasma cells. So this illustrates how we can use a common inductive site with a mucosal vaccine platform, for example, that's administered intranasally, to elicit protective immune responses, immunogenic immune responses at multiple different effector sites. Okay, so that sounds great. Why don't we have all these options for mucosal vaccines in the clinic? I think right now there's only really one that we can receive routinely. Why are there barriers to mucosal vaccine development? Well, largely the barriers are related to drug delivery. Our mucosal tissues are very good at keeping most things out, particularly subunit vaccines. And if we administer a subunit vaccine to a mucosal tissue, it's typically cleared via mucociliary clearance or degraded because of the harsh pH, the low pH, or the degradative enzymes in the mucosal layer within about 24 hours. And so what that means is that vaccine components get taken up. And if they can't get taken up, then they can't target these key lymphoid tissues to activate an adaptive immune response. But while the mucus is very good at keeping most things out, we know that two naturally occurring proteins are actually very good at getting in. And one of those is the endogenous serum protein, albumin. Albumin is naturally transcytosed across mucosal epithelium by the neonatal Fc receptor, or FcRn. So we thought, well, what if we can use albumin as a vaccine chaperone to effectively hitchhike across mucosal barriers? So we designed a vaccine platform that exploits albumin's role as a fatty acid transporter. Because of this, albumin bears multiple lipid binding sites. So we engineered a vaccine platform that has a lipophilic tail so that we could attach vaccine cargo like antigens, peptides or proteins, molecular adjuvants to this platform, to the tail that will bind albumin to effectively hitchhike across these barriers, get taken up into mucosal-associated lymphoid tissues where our vaccine can then interact with key immune cells like T cells and B cells, especially T cells and B cells that are involved in that germinal center response where immune memories are being made, activating then immune cells to become resident in that mucosal tissue. And then particularly we're interested in secretion of protective antibodies, IgG, and especially IgA, which is the main mucosal antibody. And in the setting of infectious diseases, neutralizing antibodies, which is like the gold standard that can not only bind to an antigen, but bind a viral or pathogenic antigen and neutralize the target so that it can no longer be infectious. Okay, so we went about designing this vaccine platform initially targeted against HIV. I was working, this work started during my postdoc. I was working on mucosal vaccines against HIV in Daryl Irvin's lab at MIT. And as our antigen target, because we were focused on activating a B-cell humoral response, we selected this HIV glycoprotein, envelope glycoprotein from the surface of HIV, a model antigen, a B-cell target. I will refer to this interchangeably as GP120 or EOD, engineered outer domain. We attach this to our vaccine platform, which consists of a lipophilic diacyl tail linked to a hydrophilic polyethylene glycol that helps to solubilize this. And this makes an amphiphile tail. And again, the idea is that this will exploit albumin's role as a fatty acid transporter, bind to endogenous albumin, so albumin that's already present being secreted in the mucus for effective uptake. In the presence of albumin, the concentrations we typically see in the serum and also mucosal fluids, this vaccine exists as a monomer, so it's monomerically bound to albumin. However, in the absence of albumin in aqueous solution, we actually see this revert into a micellar form, forming about 30 nanometer nanoparticles. And so this allows for pretty efficient purification using things like size exclusion, chromatography, and characterization using dynamic light scattering, which we are looking at here, efficiently purifying these self-associated micelles. But in vivo, we don't want it to be micellar. We want it to be bound to albumin to enhance the uptake. So the first question we had was, does this vaccine platform actually enhance mucosal uptake? We tested this in mice with intranasal administration of a fluorescently labeled AMP vaccine, which we call AMP-EOD, or a soluble unmodified protein control, Alexaflor 647-EOD. And immunized with an adjuvant, and then looked at signal in the mouse's nose. So here we're looking at a region of interest on the underside of the mouse's nasal passage by IVUS. You can see that the soluble unmodified protein, there's a bit of signal here at 24 hours, but it's totally cleared by 48 hours. And in contrast, our AMP vaccine persists without any reduction in signal for 3 days. And we actually still see some vaccine signal there out to 11 days. And if we quantify this, integrate the area under the curve, then we see that this combines to give a significantly greater total vaccine accumulation in the nasal cavity from this simple amphiphile modification. We then wanted to know if this was truly dependent on the neonatal C receptor. So we repeated this immunization with an FcRn knockout group and found that indeed the AMP vaccine signal was significantly knocked down to more closely resemble what we saw with the soluble protein. So this supported an FcRn-dependent uptake mechanism. And it told us about retention, but really didn't tell us yet about uptake into the epithelium. So next we took a closer look at mucosal epithelial uptake by taking histological cross-sections of the mouse nose as shown here. So here we're looking at a 5-micron cross-section of either our soluble protein in wild type, our AMP vaccine in wild type, or our AMP vaccine in FcRn knockout mice. And you can see that at 6 hours post-administration, our soluble protein really isn't being taken up on the epithelium here. So this is the negative space, the airway, and the nasal passage. If we zoom in to this box here, we can actually see the epithelium labeled with E, the mucus labeled with M, the underlying lamina propria LP. And in fact, the EOD protein is mostly just trapped in the mucus. It's trapped in the airway. In contrast, our amphiphile vaccine is nicely deposited on the epithelium. And if we take a closer look at 24 hours, we can actually see that it's being taken up as it's being transcytosed in those FcRn vesicles here in the lamina propria. So interestingly, we actually did see, despite the total reduction in overall signal with FcRn knockout mice, we still saw deposition of our AMP vaccine on the epithelium here. And we ended up determining that this was due to a secondary mechanism of the amphiphile tail inserting non-specifically into the cell membrane. And so what we now call this is cell painting. We can use this AMP tail to paint cells, effectively painting them with multivalent antigen. You can see that via microscopy here. You can see it demonstrated via flow cytometry experiment here. We incubated our just splenicized non-specific cells with our AMP EOD versus a soluble EOD. and then washed those cells, and then came in after that with an antibody that was specific for the antigen EOD. And we'd expect to see a double positive signal if that amphiphile vaccine was actually inserting into the membrane and painting the cells. And indeed, that's what we see with AMP-EOD and is totally absent with EOD. So we think that might play a role, this secondary mechanism, in aiding in the persistence of our vaccine in lymphoid tissues. And I'll get to why we think that's important, in addition to the uptake piece. Ultimately, uptake is important because we want to target these lymphoid tissues. In the nose, the mucosal-associated lymphoid tissue is the NALT, shown here in both of these regions that we were just looking at. If we're looking at the histological cross-sections, we see the NALT as these aggregated lymphoid regions at the bottom of the cross-section, at the bottom of the airway. If we're looking at the ROI from the IVUS images, that's actually not showing up here, but there's actually two little strips in this region of interest here. And the NALT's really cool, and other mucosal-associated lymphoid tissues are really cool because you actually see this organization of B cell zones and T cell zones, the microarchitectures that we also see in lymph nodes. And again, this is important because these organizations of immune cells in the germinal centers serve as a training ground for a high affinity antibody response. So there's a lot going on this slide, but the main takeaway is this is where antibody responses are trained. This is where immune memories are made. It's where we want to target our vaccine. And we found, in fact, that our amphiphile vaccine, because we saw enhanced uptake in an FcR-independent manner, we also saw that correlated with enhanced germinal center B cell activation and TFH activation in an FcR-independent manner. So here we're looking at antigen-specific germinal center B cell expansion, total germinal center B cell expansion, and T follicular helper cell expansion. And you can see enhanced with the AMP vaccine, pretty much non-existent with the soluble protein because it's not getting taken up, and also abrogated with the knockout mice. And this activation of the local germinal center response ultimately led to enhancement of systemic antibody responses. So we're looking at serum IgG as a marker of the systemic immune response, but then also excitingly, significant enhancement of mucosal antibody responses, in which case we're mostly focused on IgA. So here you can see out over 35 weeks, about 1,000 to 10,000 fold increase of serum IgG titers and 1,000-fold increase of vaginal and fecal IgA titers from our AMP vaccine. And we're looking at these antibody responses as the genitourinary mucosa as the clinically relevant site of transmission for HIV. And in fact, over 1 year post immunization, we still saw the presence of antigen-specific IgA secreting plasma cells in this mucosal tissue, in the vaginal tissue, in numbers that mimicked what we saw in the bone marrow, which is where we'd expect those plasma cells to go rest. So that was pretty exciting, indicating that we had this long-lasting humoral response. And I want to emphasize that this is striking because one of the big challenges with mucosal vaccines, particularly mucosal subunit vaccines, is typically we don't see robust immune responses activated, and typically they're not long-lasting. So here we're seeing super robust, very in mice long-lasting immune responses. Also important to note that this response was not seen when we immunized parenterally with the same vaccine. So here we compared an intranasal immunization to a scruff subcutaneous injection. And while we still see enhanced serum IgG responses, we no longer see any significant vaginal or fecal IgA responses with that scruff subQ injection. We've also tested this with SARS-CoV-2, so seeing if this platform was amenable to a different antigen, activating immunity at different mucosal sites. And so we swapped out our HIV GP120 antigen with the SARS-CoV-2 receptor binding domain, incorporated it, which went pretty smoothly actually at the time, into our amphiphile platform, and then looked for antibody responses, not only now in the genitourinary mucosa, but more clinically relevant sites like the saliva, the nasal tract, and the bronchoalveolar lavage fluid as an indicator of the lung antibody response. And here again, you can see significant enhancement, both IgG and IgA from our AMP-RBD vaccine compared to RBD. This is at 6 weeks, so 2 weeks post-boost. Testing this in the SARS-CoV-2 setting allowed us to look at that additional metric of functional immune protection, the gold standard response of neutralizing antibodies. And this was really exciting because not only did we see over 1,500-fold enhancement of neutralizing antibodies in the blood from our AMP vaccine, but we also saw over 50-fold enhancement in mucosal tissues like the nose and the lungs. which, despite their effectiveness, especially as protecting against severe disease and death, the mRNA COVID vaccines don't elicit this type of mucosal immunity in the nose. And even other mucosal vaccines in development have really, to date, struggled to activate this type of immunity in the upper respiratory tract in particular. Of note, we tested this in non-human primates as well. And I think an important consideration whenever we're looking at drug delivery in a small animal model like mice and then taking it to larger models like primates. As an engineer, as someone thinking about drug delivery, we have to think about the differences in anatomy. And for example, the differences in the surface area to volume ratio in a mouse nose, which has this highly intricate architecture of the nasal passage compared to a primate nose. I think in general, we still don't have a great understanding of how drugs, vaccines are trafficked in these larger animal models. It's something that we should investigate. We should keep this in mind as we are. Nonetheless, we saw the same overall patterns that we saw in mice repeated in non-human primates, so significantly enhanced uptake of our AMP vaccine in the nasal passage of macaques compared to the soluble vaccine. And then this corresponded with a slightly more modest, but still 10-fold enhancement of plasma IgG and IgA and nasal IgG and IgA with our AMP vaccine compared to the soluble vaccine. Okay, so wrapping up the infectious disease part of this talk, I've demonstrated this strategy for albumin-hitchhiking in vaccines to allow vaccine components to cross mucosal tissues. We've tested this in the nose, the lungs. In a few minutes, I'll show you that we've also now tested it in the gut. It's a simple modification to a vaccine platform that allows this enhanced uptake, and ultimately this enhanced uptake and targeting allows enhanced germinal center responses in mucosal-associated lymphoid tissues, which is important for priming mucosal antibody responses. So how can we now use this in the setting of cancer? So recall that we started off talking about how antibodies against endogenous retroviruses that are upregulated in tumors like those found in pancreas cancers can aid in survival outcomes. They enhance the responsiveness of immune checkpoint blockade, and this has been studied in both mice and humans now. And so we wanted to see if we could activate this type of antibody response, now with an AMP-IRV-K vaccine. And we wanted to activate this germinal center response in one of these lymphoid hubs that sits along the shared axis between the pancreas and the gut. And so lymphoid tissues to target with our vaccine along this axis include the first draining lymphoid tissue after gut uptake, which is the gut-associated lymphoid tissue, the GALT, or the Peyer's patches, mesenteric lymph nodes, and other peripancreatic lymph nodes, meaning the pancreas draining lymph nodes, and tertiary lymphoid structures, those field hospitals. And again, Noah Sinclair is the PhD student who's been working on this. And so we set out first by making a model AMP vaccine. And we wanted to use here a model antigen that more closely mimicked what our ideal goal antigen is in the end. Our goal antigen to make an AMP-IRV-K vaccine is an endogenous retroviral glycoprotein from IRV-K that's upregulated on pancreatic tumors. As we're working on that, we first started by making an AMP-GD vaccine using glycoprotein D from the herpes simplex virus, because it's about 40 kilodalton, roughly twice what we were using with the receptor binding domain in GP120 from HIV. So we wanted to use an antigen that had a more similar molecular weight, just to make sure that we could incorporate it into our platform. And we expect that the trafficking behavior we see with that would more closely mimic what we see with the IRV-K. We've been investigating two things, namely to try to target lymphoid tissues along this gut-liver axis. One is engineering formulations to enhance the stability of our AMP vaccine in the gut. And secondly is looking at multiple different routes of administration in the off chance that we can't achieve oral uptake with our AMP vaccine. So some of these stabilizing formulations, which I'll talk more about in a few minutes, are using a water and oil and water emulsion to stabilize our AMP vaccine. But oral administration is well motivated for an AMP vaccine because we see that FcRn is widely expressed in mouse intestines. So for instance, here's a histology, H&E, showing FcRn and nuclei compared to an isotype control. So we know FcRn is expressed in the gut, and that motivates us to use this strategy of FcRn hitchhiking in the gut for enhanced uptake. Initially into the gut-associated lymphoid tissues. But a few things were unknown about this. This was our first time testing oral administration of an vaccine for uptake in the gut. We didn't know how stable albumin would be, endogenous albumin in the gut. We didn't know whether we'd need to pre-incubate first with albumin, exogenous albumin to aid in that chaperone's function. We also decided that maybe we should try using lactalbumin. This is a lactose protein or a protein found in milk. So as a dietary protein, we thought maybe that would exhibit enhanced stability, and yet it still has some of those fatty acid binding domains that albumin has. And then I also mentioned the water emulsion that we also pursued to try to encapsulate our AMP vaccine into this lipid nanoparticle. And the reason for exploring these formulation strategies is because in vitro digestion assays, we found that our AMP vaccine, as well as the soluble protein, was still susceptible to digestion and degradation in stimulated gastric fluid. So here you're looking at basically binding of our AMP GD and GD significantly abrogated from exposure to stimulated gastric fluid. And one thing to note is, why would we pursue a water emulsion and incorporation of our amphiphile into this type of nanoparticle? This is motivated by the natural uptake mechanism in the gut of lipophilic particles via chylomicrons. We thought that our AMP vaccine, if it's incorporated into a fatty acid particle or a lipophilic particle, it could be taken up via that same mechanism. Okay, so we first investigated this again using IVUS, in vivo imaging, which looks like the stars didn't carry over, but you can see here that there's a significantly enhanced uptake in the gut from oral administration of AMP-GD compared to GD. With AMP-GD alone, AMP-GD plus albumin, and that significantly exceeds that of what we saw with soluble protein. This is looking at uptake in the small intestine by IVUS. We looked at signal in the stomach, the small intestine, and the large intestine. But as the small intestine is the site where the galls and underlying Peyer's patches are located, we were mostly focused on that for uptake. And then we did multi-photon microscopy, 2-photon, to look more closely at vaccine signal on the interior of the intestine. And here we were pretty excited to see that comparing AMP-G to GD, we saw significantly enhanced signal, suggesting that indeed the AMP vaccine is being retained in the gut. It would suggest that it's being taken up. And here's just a closer look at these two images. But because of some of our concern about the stability still, like we're seeing uptake, but we're still in the process of determining if the antigen that's in our AMP-GD vaccine is stable and still retains its immunogenicity and epitope binding ability. And so we're considering other routes of administration in tandem. Along with administering orally, we're investigating intraperitoneal injection, IP injection, sub-Q injection as a control, and also actually intranasal immunization. If you recall from my earlier slides, our intranasal immunization with AMP-EOD activated significant fecal antibody responses, which would suggest a humoral immune response in the gut. And so why the IP injection though? What's motivating this? Well, in fact, we see that many current pancreatic cancer and diabetes treatments are administered IP. We thought maybe there was a reason for that if we look more closely at the drug delivery, including with our AMP vaccine. And so here I want to mention both a few of our studies and a few studies that we've seen from others that show that administering IP can actually lead to enhanced uptake in the mesenteric lymph nodes and other gastric-associated lymphoid tissues. So here it's worth mentioning that in addition to enhancing uptake across mucosal tissues via FcRn trafficking, Our AMP vaccine also can exhibit enhanced uptake in lymph nodes following a sub-Q injection via this same mechanism of albumin hitchhiking, but here instead of active transport via FcRn, it's binding onto albumin and trafficking just because its molecular weight is bumped up and because albumin traffics to the lymph nodes. So this is an attractive strategy, especially if we're trying to administer cargo that is really small, like peptide antigens, small protein monomer antigens, molecular adjuvants, which typically, if you inject those sub-Q, they preferentially drain into the blood and be cleared rapidly. But here, because they're binding to albumin via this AMP tail, they're trafficking to lymph nodes, concentrating in lymph nodes, enhancing the immune response, as well as reducing off-target systemic exposure. We see that if we administer our AMP glycoprotein, via sub-Q injection, we see significant targeting to the draining inguinal lymph node from just a tail-based sub-Q immunization, quantified here with our IVIS signal, and that corresponds to an over 100-fold enhancement of serum IgG antibody responses. And again, this is because albumin, via its molecular weight of around 67 kilodalton, that bumps it up to that bumps up our binding antigen to a molecular weight where we'd expect to see over 90% retention and recovery in the lymph. Okay, so serum antibodies are great, but why are we now using an IP injection? Where does this come from for a mucosal antibody response and motivating this particular work? This stems from other work that we did. Sorry, looks like there's some formatting conversion errors here, but some other work that we did trying to activate humoral immune responses in the genitourinary tract via parenteral immunization. And we found that if we could, with an injection, target lymph nodes that drain the mucosal tract, for example, in the case of a genitourinary immune response, we were trying to target the iliac lymph nodes, which drain the female reproductive tract. We found that we could actually activate this type of IgA mucosal response. Here we investigated different routes, tail base, inner thigh, foot pad, tail, sub-Q or intravaginal administration with our AMP vaccine. We found that the tail sub-Q route of administration led to the greatest concentration of our vaccine in iliac lymph nodes. And then this corresponded to increased activation of germinal center B cell responses in the iliac lymph node and systemic IgG responses. But more importantly, this actually led to activation of fecal and vaginal IgA responses. So it demonstrates that we can actually use a parenteral immunization strategy by selectively targeting particular lymph nodes that drain a mucosal site to activate mucosal immunity. And motivated by this work, as well as that by some others, this was actually a postdoc lab mate of mine who showed that with another vaccine strategy, if you could target the mesenteric lymph nodes via an IP injection, that you could activate gut humoral immunity. This motivated us to include IP injections in our investigation, kind of as like a backup, a fail-safe to make sure that we could activate responses, even if oral uptake and transport didn't work. And we are excited to see in an initial pilot study that by IVIS, our AMP-GD actually exhibited enhanced uptake in the pancreas compared to our soluble GD. And then we actually saw activation of germinal center responses in the gut-associated lymphoid tissue, both from IP immunization, IP as well as oral administration with our AMP-GD vaccine. So here we're looking at total germinal center B cells and antigen-specific germinal center B cells. Over here, we're looking at the oral route, and namely, we see significantly enhanced activation in the AMP-GD oral route compared to the soluble GD. So from here, we're excited we've kind of laid the foundation with some preliminary data to suggest that we can use this vaccine targeting strategy to achieve enhanced uptake in gut lymphatic tissues. We're targeting these gut lymphatic tissues to activate germinal center responses and hopefully engage a humoral immune response targeted against ERVs that are upregulated on tumors. From here, we're continuing to evaluate uptake in these key lymphoid tissues along the gut, liver, pancreas axis, evaluating germinal center B cell activation in these key lymphoid tissues, ultimately with the goal of looking at downstream responses in natural killer cell, NK cells and CD8 T cells to see if we can activate an anti-tumor response via ADCC, antibody-dependent cellular cytotoxicity. We're also working on developing an ERV-expressing model system in KPC and a KPC mouse model of pancreatic cancer, both in collaboration with Lumansky and Paolo Provenzano. And this organoid tumor model will be expressing HERV-K antigens to look at targeting in a in vivo tumor model system. So overall, I've told you a story about how we can use albumin-hitchhiking vaccines to employ ancient immune memories against cancer, targeting these ancient ancestral retroviral glycoproteins that are upregulated on tumors, using a targeted hitchhiking mechanism with an amplified vaccine to activate humoral immunity and engage this humoral immune branch to activate anti-tumor immunity. I think we're really excited to investigate this in pancreatic cancer and other cancers characterized by cold tumors. With that, thanks so much for your time. I'd like to acknowledge my lab, my PhD students, our undergrads, our funding sources, as well as our collaborators, and particularly Paolo Provenzano, Lumansky, and Brandon Moriarty for this work. Thanks so much.
I have microphones for anyone who has questions in the room. We'll start with Heidi.
Go ahead. Yes.
If I wanted to use
I'm going to give you a microphone.
Okay, thank you. If I wanted to use FcRn to go across to a mucosal membrane, first thing I'd think about is to use an antibody, particularly an IgA, because that would shuttle across and might be trafficked to some dendritic site. Have those been tried?
Clarification question, IgA or IgG? So IgA.
If you're using IgA, you'd have a better shot at it trafficking to a dendritic site, because it's designed to bind a virus and take it to a site, and so if I was choosing between a G and an A, I'd go with the A, just because that might be more natural. But normally the FcRn shuttles antibodies and the things bound to them across membranes.
Yeah, that's a great question. So IgA, we don't see IgA binding FcRn as far as I know, but we see FcRn binding IgG. And so, why not just make an IgG Fc fusion, like conjugate this protein directly into that, or engineer.
Make it specific.
Sure.
That might work better.
Yeah, and so our rationale for using a conjugate that can non-covalently associate with albumin and dissociate is that we actually hypothesize that this leads to better targeting of the lymph node, because after it's taken up, it can dissociate lymph node or mucosal associated lymphoid tissue, because after it binds to albumin and is taken up, it dissociates. And we see concentration of our vaccine in the lymph node or the underlying lymphoid tissue, and we do not see significant see a little bit of drainage to other lymph nodes along that chain. For example, cervical lymph nodes following intranasal immunization, mesenteric lymph nodes in the gut. But we don't see it ending up in like the spleen or the liver or these other filtering organs or general systemic dissemination. In contrast, if you were to have an Fc fusion, like an IgG fusion, or engineer this antigen into an IgG or IgA, you'd expect that to continue circulating with the antibody, right? Like FcRn, it recycles IgG so that IgG concentrations in the serum remain high.
It certainly in the placenta, it goes both ways.
What's that?
In the placenta, for sure, it goes both ways. I don't know about the mucosal membrane, but unless there was something specific to only makes it go out, which doesn't really make sense.
Right.
It probably goes both ways. Half goes out, half goes in.
Yep, that's true. The reason we wouldn't want to have our vaccine continue circulating with an IgG is because we think that would not lead to as good of an immunogenic response. It would be more likely to lead to a tolerogenic response, because it's then just going everywhere. You're having systemic exposure. So more likely to potentially be tolerogenic, and or more likely to lead to some off-target effects, in contrast to just concentrating and kicking off in that local lymph node. So that's something we're investigating right now. We're comparing some of our vaccines with an FC fusion to see if this trafficking behavior and the local targeting and retention is actually aiding in that immunogenic response.
Okay.
Does that answer your question?
Oh, yeah, I I'm still believe that if you have it on an immunoglobulin, it's more, or particularly an IgA, it would be more likely to traffic to a dendritic immune responsive site, because that's nature.
Yeah. Yeah. And I think then it would continue circulating though, right? But
Well, I don't know. I don't know enough about the gut to know whether it would
I think
traffic or not.
Yeah, I think it's an interesting question, and it's a good question. So we're hoping to find out more with some of our trafficking studies right now. It's a particular interest of ours in understanding how the different kinetics and pharmacokinetic behavior of these vaccines in mucosal tissues can lead to activation or tolerance. So that's a key one, that continuing circulation or retention.
Let me change the subject a little bit. So this was fascinating. I thought that the confluence of these areas that you've created is really exciting. So thank you for the talk this morning. I was really curious about your choice of ERVs as a target. So, we've accumulated these sequences, maybe 10, 15% of our genome is ERVs, and it makes me think of a famous Star Trek episode where an alien said that humans are ugly bags of mostly water, but I think when you think of microbiomes and ERVs, maybe we're just ugly bags of mostly microbes, right? And as our immune system develops, though, some of these things are going to be expressed, and I wonder if you've looked at which ERVs are expressed in different tumors. Some of these data may be available in TCGA. I've never actually looked to see if there's data on that, and then tolerance. So there's bound to be really, really strong tolerance against some or all of these ERVs. And so do you have a strategy that would break tolerance? Or have you selected an ERV where you actually know is always silent, but it's expressed in these tumors and therefore would actually be immunogenic?
Yeah, that's a great question. So we've seen this is something we're still currently working on. It's kind of the stage of the project where we're at in determining the best ERV target and starting to develop the organoid model. And I'll also say now that it's not my area of expertise. We are working with Lou Mansky on this and relying a lot on his expertise on the endogenous retrovirus side. We do see significant upregulation of endogenous retroviruses in pancreatic tumors, as well as other cancers like of the ovary, lungs, colon cancer. And I'm going to hold back on giving you a number right now, but it's the percentage of expression there in contrast to other tissues is quite high. It's drastic, such that we would think we'll have significant on-target targeting and insignificant off-target. Sorry?
Same words or different?
It varies slightly for the cancer. Again, something we're still looking at, but one thing I didn't talk about much today is that we're co-administering these as a subunit vaccine with an adjuvant in all cases when we're activating an immune response. And so that's our method for overcoming tolerance, is administering strong adjuvants. A few of the ones that we're looking at are cyclic dinucleotides, a sting agonist, an amphiphile conjugate of CpG oligonucleotides, so that we observe the same albumin-hitchhiking and uptake across mucosal tissues as the antigen conjugate, and also another nanoparticle adjuvant called saponin MPLA nanoparticles, which via TLR4 activation is a really good B cell adjuvant. And so by tuning some of those adjuvants that we're co-administering, both the dose optimization as well as screening for activation of different immune responses, we're trying to overcome tolerance that way. But I do know that ERV expression and immune responses are associated with some autoimmune disorders like lupus, SLE, and so that's definitely a consideration. And I would welcome any additional discussion or, advice for people who have expertise in the endogenous retrovirus area.
So your lipid antigen constructs, your cartoons were showing them forming micelles in solution. But then when you mix with albumin, it looked like it was more of like a one molecule of your construct to one molecule of albumin. So what are the structures that form when complex with albumin? And does it matter?
I'm not sure how quickly I'm not going to go back right now.
Does albumin break the micelle?
It does break the micelle. We see, so if I do go back, I'll talk as I'm doing it. When we characterize this by dynamic light scattering, as well as size exclusion chromatography, in aqueous solution, we see this formation of micelles as shown here by SEC and DLS. But for example, if we run this through a size exclusion column and pre-incubate with albumin, the AMP-GP120 then it traffics with the soluble protein. So it definitely disrupts the micelles. And we've done studies where we've added in different concentrations of albumin to see at what point we see micellar disruption. It is concentration dependent. It's concentration dependent on both the AMP vaccine because there's a critical micellar concentration, at which point it needs enough to start forming micelles. And then there's a critical concentration of albumin, which we've related to literature values reported in the serum and the mucosa. And we expect it to be mostly monovalent in biological fluids.
Hi, lovely talk. I was thinking about biologics, and is the nasal route equivalent in humans to like an inhaler, like delivery to the lungs, or is it more nasal based? And is there a limit to the size or complexity of molecules? So a lot of our biologics that we use to activate immune cells have FC domains or albumin incorporated into them. Could they be used to deliver drugs to a lung cancer setting more efficiently, or would they be broken down or?
Yeah. To just making sure I understand your question. So first it was the intranasal route and whether it's also targeting lungs just nose. And then second was if we could incorporate other types of cargo.
Or if you were thinking about doing this with a larger, more complex cargo. I'm thinking of the trikes, which have FC domains or could have like albumin sections in them. But would they be broken down in this transport route? Is it something special about the very small?
Yeah, and okay, so
all right, making you hypothesize.
Yeah, and I just to clarify, so that not conjugating those to an amplifier, but using them in their native form via their FC binding domain. Yeah. Okay. Great question. So in this mouse work, as well as our non-human primate work, we were trying to specifically target the NALT and keep the vaccine as much as possible in the upper respiratory tract. Typically, if you're doing intranasal immunization of mice, like their nostrils are very small. They have small noses. It tends to go everywhere. And if you're like inoculating for influenza or something, you tend to just like hold the mouse upright, administer it, and it just drains into the lungs. Here we had the mice in a supine position on their backs, and we had administered really slowly, drop-wise, to keep it retained in the lung. And then we had them anesthetized and then kept them anesthetized for a few minutes afterwards in that position to enhance the absorption in the nose and minimize drainage into the lungs. And we did some IVUS studies early on of the trachea, of the lungs, of the nose to maximize the signal staying at the top. We did the same thing in rhesus macaques. And the reason for that, again, was targeting the NALT. We didn't want to target the lungs here. And if you were to translate, because we wanted to specifically activate the NALT for its role, especially in as an inductive site for priming immune responses, also in distal sites like the genitourinary mucosa. So what originally motivated that. If you were to translate this to humans, you could achieve a similar response by formulating an aerosol delivery, like an intranasal, inhaler, and by adjusting the particle size, you could control the deposition location, right? Particle size and velocity. So can try to like target the droplets to land in the nose versus other areas in the lungs. When we've investigated lung targeting with this. We actually do an intratracheal administration to make sure we're just getting into the lungs. And we see activation there of mesenteric lymph nodes and activation of B cell and T cell responses. I didn't talk about that today, but okay, so hopefully that answers the first part. Sorry, I probably went off a little too much on that. Secondly, do I think that other types of cargo would be broken up via this transcytosis method? I don't know. I don't think so. I mean, like with FC fusions, you see efficient uptake with this, with FcRn. I do know that the amphiphile vaccine conjugate, the amphiphile does stabilize our antigen slightly. And so that may play a role for some other types of molecules or drugs, but I think it's worth exploring, and other groups are exploring this for other types of constructs too.
All right, we'll take our final question here.
This was a great talk. Thank you so much for talking about your work. I have a question about once the vaccine, so say that you are able to demonstrate that you are getting that activation in those tertiary lymphoid structures. An infectious disease model is very different than a cancer model.
Mm-hmm.
So how do you imagine that this is going to overcome those physical barriers that are present in those immunologically cold tumors, whether that's just extracellular matrix components or even that immunosuppression? So even if you do get that activation, how are you still going to then get the vaccine?
I'm so glad you asked that because I didn't talk about it very much here, and that was an omission on my part. So we are going to test this AMP vaccine alone, like with an adjuvant to overcome tolerance for activation of GC responses and humoral antibody responses. But we don't think this is going to be effective as a vaccine alone. So we'll be testing this in combination with other therapies, anti-PD-1s, anti-CTLA-4, even though that's less likely to work, but checkpoint blockade and KRAS inhibitors, and potentially down the road, other CAR T-cell therapies. But definitely we think that this will play a role in waking up an immune response by engaging the B cells and antibodies, but it won't do the trick on its own. And so we will need these other therapies in combination to try to overcome some of those immunosuppressive aspects of the tumor microenvironment, pull in these other important immune cells. But we do think that like the local priming and specifically the antibody response will help with that. Does that answer your question?
All right, let's say thank you one more time to Dr. Hartwell.